One concept that is key for our understanding of evolution and development, and just evolution in general, is an idea of modulars or modules, and then how animals can be viewed as being made up of these various different components. So, modularization is an idea that extends not just into biology, but we see it in many facets of human activity. So, for instance, architecture is often based on modules, right? We have these repeated units that we use over and over and over again. And sometimes they're very similar, and other times they're quite different. So, for instance, if you build a apartment building, right, you're going to have these individual modules where if you went from one to the other, they may look nearly identical. But if you're in a home, you have rooms that, although they share some characteristics, they have unique and different characteristics when you look between them. So a bedroom is different than a bathroom, and a bathroom is different than a kitchen. Now, it turns out when we look at animals, we see something quite similar, right? So one great example of this is segmentation. This is readily apparent in both arthropods and in annelids, right? Segmented worms. So these are two really important groups of animals. Arthropods are the most diverse group. Annelids, we typically think of them as earthworms, but there's a wide variety of marine worms, including like this polychaete. And each of the individual segments originally, ancestrally, when they first evolved, was nearly identical to all of the others. But since then, individual segments have evolved. And in some organisms, this is only a little bit where we get, much the same from front to back. But in others, there's been a huge amount of differentiation between segments. So each unit, each segment can be thought of its own little individual module. Okay? So much like rooms in a house or units in an apartment building, right? These are built independently to some extent, but are all based on the same basic ground plan. Now, to some extent, humans might be imposing our own kind of worldview on animals, trying to make them convenient, but there really is, for almost all examples of this, a real biological, it's not just us kind of trying to break things down to understand them. It's really a important idea for understanding how these organisms have evolved. Okay, so many modules exist, and we see them in all animals to one extent or another. You might make an exception. Some of the simpler animals, like Placozoa in particular, maybe even the sponges, you have a harder time looking at modules, but even then you could go down to the cellular level, right? Certainly a cell can be thought as a module, and then differentiation of those cells is what is one of the fundamental differences of animals. So if we have to go down to the cellular level, we can still see it. But most organisms, there are layers upon layers of different modules. So another example are the vertebra in animals, in chordates, right? So each individual bone is somewhat similar to the others in the entire vertebral column. So the cervical vertebra, the thoracic vertebra, right, including the sacral, the lumbar, and all those different parts, and then the tail vertebra, they all have some features in common, but they have differentiated to take on unique features, right? So we have the thoracic vertebra, some of which don't have ribs attached, some of which do, right? We have the cervical vertebra that help to connect the thorax to the head. And each of those can be looked as an individual module with diversification on the basic ground plan. And that gives us one of the key ideas of modules. We'll define that a little bit more, but it's basically we start with a basic underlying structure, we multiply it, so we get more of them, and then we can diversify once we have multiple repeated modules. The forelimbs and hind limbs of vertebrates are also modules, right? And you can see some similarities between them there. Anciently were very similar, but have diversified in many groups. Although some groups still have a very simple plan with forelimbs and hind limbs being much the same. This bat obviously has some quite distinct differentiations between forelimbs and hind limbs. So teeth, right? That's another module. And many structures can be viewed through this lens of modularization. Another great example is the insect wing. So we've got veins that make up the structural components with cells in between them, and those are repeated structures that we see over and over and over again. The last example here of modularization more at the molecular or at the morphological level that we're going to do is the limb itself. So the limb itself is a module, and within that you can see subunits, or maybe call them submodules, right? Maybe the best example of this are the digits. Now this drawing is a diagram of an ancient tetrapod vertebrate, one of the earliest tetrapod vertebrates. And notice it has like 8 digits, 1, 2, 3, 4, 5, 6, 7, 8, yeah. 8 separate digits, each with individual phalanges, right, that themselves can be looked at as modules, but the digit itself is a module, right? Now, the ground plan, this was a very early tetrapod vertebrate that eventually went extinct. The ground plan for all living tetrapod vertebrates is 5 digits on each limb, but that itself the number has been modified. We don't see any living organisms with more than 5, at least species, let's say that. We don't see any living species that have more than 5 digits, although occasionally there are rare mutants in different groups, including humans, that have supernumerary digits. But for some reason, they never seem to be an adaptive benefit. So all species have 5 or less because many species have lost digits on their forelimbs or their hind limbs. But again, great units, modules within modules. So just be aware of these examples, right? So at the morphological level, we define a module by comparing it to other components, either in the same organism or in closely or distantly related organisms. So basically, we define a module by comparison. And if we see similarities of individual units that make up an organism, then we can define it as a module and begin to investigate how they originated and perhaps how they have differentiated from one another. And so key to our understanding of modules is this idea of homology. And in fact, we're going to introduce a different level of homology here. So homology, remember, is just similarity due to inheritance from a common ancestor. So if I looked at this ancient amphibian forelimb and compared it to the forelimb of a bat, I'm going to see some similarities. So notice that there is a large bone that is most proximal to its connection to the thorax. We have the same thing in the bat, different shape obviously, but a single long bone. Then there are two bones, and then there is a number of digits. So the wrist bone in this is not complex. So we have few bones here. In fact, it looks like 0 in this one making up the wrist, and a more complex form of bones making up the rest is here. So that would be a difference in the modules of the bat and the early ancestors. But the key to this is homologizing, that we can compare this first long bone to the first long bone that we see across tetrapod vertebrates today. Okay, so that homology idea is key. Now, the more complex version here, in fact, let's add to this, is this idea of serial homology, or saying something is serially homologous. And this is a really key idea for the origin of different modules. So let's define what we mean by serially homologous. So originally, when modules begin to be repeated, almost certainly, at least initially and maybe even for long periods of time, those repeated modules are nearly identical to one another. And we see this over and over again. So the early arthropods, every segment, if you look at a trilobite, every segment that has a leg on it looks much like the segment in front of it and the segment in back of it. And some arthropods have maintained that distinction. So I think I've got images here. So here is a close relative of the arthropods, velvet worms, Onychophora. It has a leg on every single segment. The leg in the middle looks much like the leg in the front, looks much like the leg in the back, right? And we see this in other arthropods like the centipede, right? Front legs, hind legs, pretty much all the same, okay? So initially, we get this idea of a repeated morphological unit that are pretty much identical to one another. And so when we have a repeated unit that is identical to the one in front of it and behind it, we call that thing serially homologous. So unit 1 is homologous to unit 1 in another species, but it's also serially homologous to unit 2, 3, 4, and 5 in its own body. And we could do the same thing for vertebrates, right? The forelimbs of vertebrates are serially homologous to the hind limbs in vertebrates. So we can productively compare the forelimb of a bat to the hind limb of a bat, or even the forelimb of a bat to the hind limb of a chicken, because originally they started out as this modular unit that was identical. And in the first fossil vertebrates we see, and in some vertebrates today, like a lizard, for instance, or an alligator, forelimb and hind limbs are still largely the same, maybe even nearly identical. But in many organisms, we have gone on to then differentiate these originally similar and serially homologous modules. So there's some terminology there, but hopefully those ideas are set out in your head and you can see how it's useful and valuable to look at animals through this lens of modularization. There's a paper where some of these things were outlined. I'm not having you read it for this class, but I just put that reference there, and you don't really need to go after that unless you're really, really interested in it. Okay, so we've established this idea of serial homology at the morphological level, but we want to extend this, and we're going to begin to start to do this more and more throughout the semester. We want to extend this to the genetic level, because that's the overall goal of Evo-Devo after all, is to try and connect what's going on with our genes to how we build an animal and how we make a final, functional organism. Okay, so what is the genetic equivalent of modularization? And just like at the morphological level, we can actually do this at a couple of different levels, right? So we can maybe even go all the way down to the very basic level of molecules, right? A nucleotide. Is that a module? Well, yeah, it is. Adenine is similar to thymine and guanine and cytosine. So we could look at the individual nucleotides in DNA as the most simple, broken down part of a module. But then we can extend that and say, well, okay, but we can put those together and make a gene. And that gene functions as a unit. Okay. Now, there are two ideas that I want to make sure, definitions, that I want to make sure are very clear in your head. And then I'm gonna skip the 3 criteria modules. In fact, I'll delete this. And rather than this, I think it's going to be more productive. Since I'm not having you read this paper, I'll get rid of this on the PowerPoint so you won't even see it on the version I post online. Rather than that, let's make sure that these 2 definitions are very clear in your head. Okay, so you should know pleiotropy. And then the other thing I want to make sure is polygeny. And these are also important at the genetic level when we're talking about modularization. Okay, so let's define these. Pleiotropy is where one gene has 3 or 4 or more jobs, really any more than one, and it's defined as pleiotropic. So a gene with more than one job. Now, the flip side of that, you can maybe think of it even as the opposite, if you'd like, is polygeny. And polygeny is where many different genes all contribute to the same phenotype, right? So one gene, many jobs, many genes, one job, or at least very similar. They're all contributing to the same, like building a leg, for instance. That, if you're looking at that way, that's a polygeny. And many of these modules, which are very complex, are going to have many, many genes that are all going in to build them. But we could establish homology at the genetic level. If I had a series of genes that I knew, say, built a leg, and then I saw a very similar set of genes that was building an antenna, that's a sign at the genetic level that an antenna and a leg are homologous, that they were built from that same basic genetic module. And even though they might be somewhat different today, if there's enough similarity overlapping, we can assume the same ancient origin through duplication, serial homology, and then diversification of that original plan. And so that really begins to answer this question. What are the advantages of modularization? And we're going to put 2 big advantages. The first one, which is maybe fairly obvious, is redundancy. Okay? So if you have many, many structures all doing the same function, if one of them malfunctions or gets damaged, or there's a mutation, it doesn't form correctly, then it's not as big of a deal because you have many others that can take up the slack, right? So redundancy is a major advantage of modularization. The second major advantage is it gives you a wealth of structures that can then be diversified differentially. So it is a stable ground plan from which more diversity can be built. And so that's the second advantage of modularity. And we see that over and over again. So here's some of that diversity. Here are arthropod appendages. In the scorpion, we have these front limbs that are pinchers. We have 4 sets of walking limbs. And then we have segments without any limbs on them at all. So diversification of that same ancestral plan. Some legs are diversified for swimming. Some appendages are diversified for feeding or for sensory structure. Maybe in the shrimp, there are other arthropods that are nearly as diversified. But in the shrimp, we have this incredible example of this diversification of individual modules. Now remember, in the ancestor of arthropods, pretty much every segment was the same as the front and the back. The head appendages differentiated fairly early on, but the thoracic and abdominal segments almost always just have a walking leg or a swimming leg in the early arthropods. But in modern arthropods, we have this incredible array of diversity. We have antennae, 2 sets in the crustaceans. We have these mouth parts here, the maxillule and the maxilla. We have these walking or swimming legs in the shrimp. We have these that have a gill on them and are also used for brooding the eggs. We have these appendages at the back that have been modified to make a tail. And so you can almost think of them as like the Swiss Army knife of arthropods. Do you know what a Swiss Army knife is? I'm just making that assumption. When I was a kid, I loved pocket knives. And a Swiss Army knife is a pocket knife with like 5 or 6, or some of them have like 100 different blades. You know, the one with the sawing blade and the cutting blade and the can opener and whatever. So in some ways we can look at arthropods as this multi-diversified, every module, which was once the same, now diversified. And so I can compare thoracic segment 1 to thoracic segment number 8. But to some extent, I can also compare it to a abdominal segment, right? And say, what are the similarities? They all diverge from the same original repeated module. And so this diversification of repeated modules, originally redundancy, and that's probably why most of the time mutations that increase the number of modules were kept, because you have redundancy and that provides a bit of a benefit. And then you have a open playground, if you will, for evolution to work on. More mutations can occur. To diversify them, you can keep some of the original function and then have some of the other appendages take on new function, and so you still maintain what you had before, but then you gain additional features also without having to evolve them completely from scratch. And again, I mentioned this, we can break down modules within modules. So we're going to be using insects in our lab for this class, so you'll get a little bit more familiar with them. But insects have 3 major body regions. They have a head with a number of segments, a thorax with 3 segments, and then an abdomen, again, with a number of segments. Sometimes some organisms have more, some have less. And so we can diversify the structures on these different ones. So the big module might be head, thorax, abdomen, but within each of those we have a repeated segment and a leg on some of those segments. So the leg is a module for the thoracic segments. So we have modules within modules, and we can break it down all the way, maybe even down to the cellular level or even the molecular level if we want to get very, very detailed.